Okay, so on Wednesday, we left off here talking about how bacteria and archaea can be so similar structurally while being so different chemically. And we basically addressed this question along the lines that there are certain things that all living organisms have to have, right? All living organisms have DNA. All living organisms are made of cells. They either are a cell or they contain many cells. All living organisms, respond to stimuli. They metabolize. They grow and develop. They make copies of themselves. So there are certain machines that you need to find in all living organisms. On top of that, bacteria and archaea both lack nuclei. They're both prokaryotic cells. And so because of that, the intracellular structure should be fairly similar, okay? And they have similar functional needs. Most prokaryotes also have a cell wall, a non-living structure that's deposited by the cell to provide some additional structure, to provide some additional safety for the organism. Now, in bacteria, it is made of peptidoglycan. And in archaea, it is made up of a number of different chemical materials. So it's chemically very different. Their walls are very different than bacteria. But they oftentimes build a wall for the same reason, to provide additional safety for the cell. Many of these prokaryotes, bacteria and archaea, oftentimes have flagella, which are basically machines for moving the cell. Pili, which are tubes for transferring information or attaching to an object. And then plasmids are little pieces of DNA outside of the main chromosome. And so many bacteria have these, many archaea have these. And it's because they both need to move. They both sometimes need to attach or to transfer information to another individual, and they both need to basically have a mechanism by which they can transfer genetic material, and that requires these plasmids. So why are they so similar structurally while being different chemically? The main reason for that is they have separate origins, separate ancestry, but similar life histories. Okay? They have separate ancestry, meaning that they don't share an ancestor that would be labeled as one or the other. They come from separate origins, but they have similar life histories, meaning that their life cycles are very similar. They have similar needs and similar means by which they meet those needs. Okay? Does that make sense? Yeah, Cameron.
So they both bacteria and archaea lack internal membrane bound structure.
Yes, and lack nuclei.
Okay, yeah. But it says they have the plasma membrane. So is that because it's a...
So they still have that outermost membrane. They don't have any internal
They
membranes.
have a cell membrane?
Yeah, that would be the plasma membrane.
Oh.
Yeah, plasma membrane, cell membrane are synonymous. Yeah.
For the pili?
Yes, pili?
The pili? Sorry.
Yeah, single would be pilus.
Okay. What were the two?
Yeah, for transferring information or attaching to objects.
And
I mean, either way, if you're going to transfer information, you attach to another cell. Either way, you're attaching.
And the plasmid also transferred?
No, plasmids are what get transferred. They're small pieces of genetic material. Like, let's say you were a bacterium and you are resistant to some antibiotic. Okay? And what makes you antibiotic resistant is some gene that you have. And you really want to give that very helpful gene to other bacteria. The pilus would be the structure that attaches to that cell and facilitates that. And then the plasmid would be a small piece of DNA including that gene. Yeah, Levi.
So is it like a spermatozoa, or is it different from that?
No, it's attached to the cell. It's not like a separate cell that is sent out. The spermatozoa is a separate cell with a flagellum and it'll swim places. This is almost like a tube. Like, have you ever seen? Yes. Have you ever seen? Oh, go. You had another question.
It's like a tube. And
Yes.
Plasmid. goes through it.
Yes.
But is it genetically, is it like kind of a spermatozoa, like it's a process? Is it sexual reproduction or is it?
Yeah, I mean it falls under the category of sexual reproduction, but it's so completely different than that. I mean, it would be in a different category. Yeah. Any other questions? All right, so some of this, we've seen these shapes in lab. You have coccibacteria, you've got bacilli or rod-shaped, and you have spirili or spiral-shaped bacteria. And these are shapes that you find in both bacteria and archaeans. Here's a typical prokaryotic cell. Notice the genetic material is not held within a nucleus, right? They are prokaryotic, they lack nuclei, and they lack other membrane-bound structures inside of the cell, but they do have a cell membrane or a plasma membrane. And then in this case, you've got a cell wall, this non-living structure secreted by the cell, and in this particular bacterium, you also have a capsule surrounding that, probably made of sugar that traps other materials. And then here you have the flagellum, the machine used for what? Movement, and that's something that you oftentimes find in both bacteria and archaea. Yeah.
So a lot of cells don't have organelles, but why do prokaryotes have ribosomes?
Because they still have to make proteins, and we'll talk about that later on. So the ribosomes are where the translation happens. Remember, we had the diagram up, and we translate from RNA to protein. That happens on ribosomes. Yep. So because you have to be able to translate from the code to the machine that actually carries out that code, you have to have ribosomes in order to do that. Yeah, it's a good question. All right, so here's the idea of bacteria and archaea. So here we have bacteria, here we have archaea, noticing that they come from separate ancestries. And so regardless of the philosophical commitment of the scientists, most people would argue that there is a separate ancestry to both these groups to get to, if you do root all life back to a single ancestor, a single survival machine, you have to go back to the ancestor of all living forms. And so the reason why they're so different chemically is because they have separate ancestry. But the reason why they're similar structurally is because they're both prokaryotic and therefore have to carry out all of the activities of life in a single cell. Okay? All right? And then there's some different phyla within here, but we'll save that for the next slide. All right? Any questions? Okay, I'm going to show you a table, and this isn't going to happen often, but what I'm going to do with this table, you are not going to like it. Okay? Now, I'm not trying to tell you that you disliking something I do is not going to happen often. That's going to happen all the time. But what specifically I'm going to do with this table that you're not going to like is not going to happen often. So I'll switch up. I'll vary the things that I do that cut you deep inside and upset you. All right? Okay, so here's a table right out of your textbook. Okay, it's really hard to read on a slide, but that's okay, because we're not actually going to go through it. However, you have to be prepared to have an assessment that gives you a group, this is alpha proteobacteria, and gives you an example bacterium, and you would need to match the two. Okay? There's another column of this table that tells you basically representative organisms and then gives you a picture of those. So you need to be prepared to be able to have a matching question where it gives you a taxon, alpha proteobacteria or beta proteobacteria or gamma beta proteobacteria or epsilon proteobacteria. You getting the picture? And then like an example bacteria. Now I'm not going to use any that aren't on this table, so I'm not going to use like surprise, here's an extra bacterium that wasn't in this table that you're responsible for, but I'm going to assume that you take this table and you familiarize yourself with it. Now here it gives you kind of some information about the group. That's really helpful to know to help you remember this, but you won't be assessed on that. Like you won't be given a question where it'll say describe alpha proteobacteria. Okay? But you will be given a question where it says alpha proteobacteria and then it has rickettsia and you've got to match the two. Okay? Yeah, Levi.
So is it the same pictures that are in the table, or is it
Oh, it won't be pictures. It'll be names.
Oh, okay.
Yeah, it'll be the names of the bacteria.
Yikes.
Yeah. Well, pictures would be, I think, more difficult if I just gave you a picture of a bacteria, because you'd have to memorize the names of all those bacteria, what they look like, and what taxon they are. Yeah. I told you you're not going to like it. Not going to like it. And again, I'm not saying that you won't often have things that you don't like, but it won't often be like this. Oh, and here's a continuation of that table, and here's a continuation of the table. There are, I think there are maybe 13 taxa between bacteria and archaea. So it's a lot, but it's not insurmountable. And really, I mean, you don't have to worry about it until exam 1, which is really long from now, 2 weeks from today. Yeah.
So we need to know the class, and do we need to know the names and the picture?
No, just the names. You won't be held responsible to be able to identify what the bacteria is by image.
So just the first 2 columns?
Yes. Yep.
Are we going to be getting into that later? That kind of stuff? Or
Getting into what? I'm sorry.
identifying things by looking at them? Is that something you're going to do?
Not really. Not really. Yeah, it's a good question. All right. Any other questions? I told you you're not going to like it. I'm sorry. It happens. Yeah.
Said exam 1, not.
Exam 1. Yeah, which I'm pretty sure is 2 weeks from today. I'm pretty sure that that's how it works. Next Friday is quiz 3, and then Friday 2 weeks from now is exam 1. All right. So here is just some of the chemical differences between bacteria and archaea. And as you look at this, I will not give you these images and have you identify it down to, I just want you to appreciate the chemical differences between the two groups. And again, that's due to separate ancestry. And then here's some more differences, but these are within bacteria. You have gram-positive bacteria and gram-negative bacteria. And you can tell the difference in the size of the wall and the presence of a second outer membrane. Again, I'm not going to give you these images and have you identify what type of bacteria it is. This will be helpful for you as you work through those taxa, right? As you work through those taxa and you identify them, because there's one taxon that is just gram-positive bacteria. And it's helpful to know a little bit about what that is, but you won't be assessed on whether you've memorized what that actually is. Not in this class. If you take medical microbiology, you have all sorts of fun with that. All right? So, on that note, since everybody's a little sad, maybe a smidgen angry, we're going to take a lecture break. Okay? And so what I want you to do during this lecture break is think back to what we talked about yesterday when we asked the question, why are extremophiles so interesting? And it's because a dominant view in science is that the early Earth was a very extreme environment, and the first life forms would have been very similar to modern day extremophiles, okay? Right? We mentioned that yesterday. And so I want you to start with that premise, and I want you to spend a couple of minutes working with those around you and come up with a list of things that would need to change in order to generate some of the other types of bacteria from that type of bacterium, okay? So if you have just an extremophile, and remember we mentioned a little bit on Wednesday about what that actually means. Emerson, you asked a question, I think, that I answered. That means like their proteins function best at certain temperatures, and that's, maybe it was over here, and that's why they oftentimes don't survive under normal conditions. So I want you to start to think through what would be some things that would need to change if you were in fact going to get all of the bacteria we have now from a extremophilic bacterium. And just for fun, also start to come up with a list of things that would need to change to generate a eukaryotic cell. Something that, unlike prokaryotes, does have a nucleus, does have membrane-bound structures, okay? And if you're like, wow, this is beyond my pay grade, we are in the process of promoting you, right? So you are taking the test for a promotion that you will get next week, all right? So take a couple of minutes working with those around you, come up with a list of features that would need to change to get all the bacteria we have from an extremophilic bacteria, and then how do you get a prokaryotic cell an extremophilic bacterium. All right? Couple of minutes, starting now. 14, 15. Focus your thoughts on So you only have one time
very thin, very basic, so you need to become like more neutral for
How do we go? What needs to happen?
The second question was essentially what's the difference between eukaryotes
Finish on Wednesday. All of it is a sheet of itself. Something is what is it? What are they?
So then also what they would need to be prokaryotic?
No, to generate eukaryotic cells as well. All right. Happy Friday again. I know it's like, it doesn't feel happy. I know it's Friday, but I'm angry and I'm usually so happy on Fridays. I don't know, maybe I'm overestimating the impact that what I just did had on you, but I don't think so. So what are some thoughts? Okay, so what needs to happen if the only type of bacteria you have are bacteria that live in the extreme environment, present in an early Earth, or on an early Earth? Okay, what needs to happen in order to be able to generate all the different types of bacteria that we have? Yeah, Amanda.
Because the what extreme environments we have today live in extremely high, extremely cold environments.
Yes.
The temperatures need to like balance out. Like they can't be too hot or too cold, and so they need to balance out to like an average temperature.
Yeah, so that's one thing so are you focusing on that the environment needs to change? And that's a key thing that's oftentimes overlooked, right? If you're going to generate all of the diversity of life from this one original form, something has to change with that environment, right? The environment has to change because if all you have is an extreme environment, all you're going to have are extremophiles. But if you want to create some diversity, you need some diverse environments. Good. Yeah, Allison, and then we'll come over to Micah.
We were also talking about there needs to be oxygen.
Yeah.
Because I think you said that most extremophiles are somewhere in oxygen.
Yes. Yeah, so yeah, what's interesting is I don't know if you know this or not. This is somewhat bonus information, but it's helpful to know. Hot water does a really bad job of holding on to oxygen. Really bad. I mean, and you're talking about like the difference between, let's say, 55-degree water and 75-degree water. They hold a dramatically different amount of oxygen. 55-degree water holds a lot of oxygen. 75-degree water holds very little oxygen. And that's only a 20-degree Fahrenheit difference, which is a little bit over 10 degrees Celsius. And if you're talking about like a 40 or 50 degrees Celsius difference, or a 70 or 80 degrees Celsius difference, the amount of oxygen you can have in water that's right beneath the boiling point of water is really, really small. Really small. So it's true. Yeah, extremophilic organisms usually live in very oxygen-poor environments, regardless of what type of extremophile they are. And the early Earth, the idea is, because the rocks look like they were made without the presence of oxygen, is that the early Earth had no oxygen. Right? It's also convenient. Anyways, we'll talk about that in a little bit. But yeah, so you need to start to actually generate oxygen, which means you need to be able to chemically develop a means by which you can make oxygen. And you all know photosynthesis really well, right? So you're like, I got it. Photosynthesis, you take carbon dioxide and you take water and you generate, so you take carbon dioxide and you take water and you generate sugar and oxygen. You're like, I got that, right? I started to roll through it and it didn't sound right, but it is, okay? And so you're like, okay, I got this, but we need to, it's not quite that simple. Photosynthesis doesn't happen spontaneously, right? If you take carbon dioxide and water, there's a ton of that in our air. It doesn't just spontaneously turn into sugar and oxygen. Be nice if it did, right? Then the air would taste delicious, right? You went on a run with your mouth open, it'd be like eating cotton candy, right? But it doesn't happen. You need machinery to make that process happen. You like that, right? Oh man, that was a good analogy. Yeah. Yeah, absolutely. You need to develop an entirely different metabolic process. Sure. I mean, yes. I mean, ultimately, if you're going to generate new materials, it has to start in the blueprints, right? If you're going to generate new materials, you need to make something novel. You need some new blueprints or instructions to make that new novel information, which is interesting. Now, these are bacteria. They are not viruses. Their genomes are pretty significant, right? A viral genome is teeny tiny. You can't pack a lot of information in it. A bacterial genome, it's significantly larger. You can pack a lot of information in there. Right? Which is why, I don't know if you know this, but we're using bacteria to actually make human proteins, right? So it's like, you have let's say you have trouble making insulin, right? And you're like, we need to get your body to make insulin. You can reprogram your gut bacteria to make human insulin, because there's plenty of space in there to actually insert human genes and get the bacteria to make those into human protein. It's wonderful. All right. Any other ideas or thoughts or comments about what would need to happen? So we need new genetic material, right? We need new codes, new instructions on how to make new machines. We need new machines that are making novel materials. We need the environment to change so that we have some non-extreme environments for organisms to start to live in. And we haven't even got to how do we make eukaryotic cells. Right? Because now you don't just need more information and new machines, but you need entirely novel structures, things that never existed before. Yeah, Micah.
I was going to say.
I totally skipped you, didn't I? I said we were going to come here, we're going to come back, and then I started asking. I'm so sorry, Micah.
Sorry, I was going to say homeostasis in the extremophile will need to change.
Absolutely. Yeah, and so this is, if it's a living organism, it maintains some form of homeostasis. Now, it may not do it like a mammal, right? Mammals, we commonly referred to as warm-blooded, which is silly because our blood's not any warmer than another animal usually. But it really just means like we maintain a narrow environmental window, right? We maintain our temperature in a narrow window. And not all organisms have such a narrow window, but they have some window that they maintain. But if they're living in extreme environments, that window is really hot, right? It's a really hot window. And we needed it to come back down to like what a typical organism would like to experience. Yeah. And you're like, I don't know about you, but I like getting into a hot pool, right? Don't do it, it's not worth it. It's like, oh yeah, 100-degree water, that's wonderful. Yeah, Fahrenheit, not Celsius, right? 100-degree water Fahrenheit, that's good, right? That's like 38 degrees Celsius. Just kind of put that in perspective. 100 degrees Celsius, that's not good. Yeah.
I don't know if this falls into the same category as mechanisms, but whatever sort of outer shell or wall or membrane they have will have to change.
Yeah.
Because I'm assuming, acetophiles or whatever it is, they don't get eaten by the acid or burned up.
Sure.
At hot temperatures.
Yeah, no, you're right. You're exactly right. Yeah, my daughter was asking me, I don't know, maybe like a week ago, like, why doesn't our stomach burn up because of the stomach acid? It's like, I mean, it does, just slowly, right? And then those cells, have just crazy, I don't know if you knew this or not, but their mitotic rate is just astronomically high. They just are constantly dividing to replace cells that get burned up. They do get burned up, just slowly, and you've got mucus lining your stomach as long as it's working properly. All right, but yeah, absolutely. Everything about their chemistry has to change. And if the chemistry's got to change, machinery's got to change. If machinery's got to change, instructions have to change. And if instructions have to change, we need some mechanism by which we get new information. It's fun. So it's a challenge, right? It's a challenge. It's not quite as simple as just saying, oh, well, they're bacteria. Of course they can develop into other type of bacteria. It's not that simple. It's not that simple. All right, so our next question. How does understanding the metabolism of bacteria help us find them? Help us find them. So I mentioned on Wednesday that most bacteria we know, or most extremophiles we know only from DNA. And actually that's true of even non-extremophilic bacteria, where we've taken like a bucket full of ocean water and then taken all of the DNA we could find in it, and we're like, this is a bacterium, but it's like nothing we've ever seen before, but we don't actually know what the bacteria looks like, or, yeah.
Did we, I missed it, I don't understand, how did we answer that, how they make eukaryotic cells? Everything has to change?
Yeah, I mean, so then on top of developing new novel machines, you also have to develop new structures that haven't existed before. A nucleus and membrane-bound organelles. And that is so impossible that the idea behind where it came from is it came from somewhere outside. Right? The structure was already in place and then entered into the cell, but we'll get there next week. Okay, because of actually making those novel structures, we have no mechanism by which we can do that from within. It has to come from without.
Is that the mitochondrial?
Yes, endosymbiotic theory. Yeah, we'll get there. All right, so how does understanding the metabolism of bacteria help us find them? So like all living organisms, bacteria metabolize nutrients, right? That's one of the characteristics of life, metabolism. And so some of these things that they metabolize: carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. I don't know if you look at those and those really ring a bell for you because you've had a previous biology class and you realize those are the most common nutrients in our bodies as well. You're like, wow, they need just the same things I need, right? We're like cousins. Except for we're not, because anyways, we'll get there. Most also need iron to metabolize other materials. We need iron for helping in our metabolism, but also for something that we have that bacteria do not. And what is that?
Blood cells?
Yeah, blood cells. We need a lot of iron to make red blood cells. This is why if you lose a lot of blood, your iron levels are really low because you just lost a lot of iron. Anyways, it's another story for another time. So we can take bacteria then and we can organize them based on where they obtain their energy, where does their carbon come from? Because they all need carbon. We need carbon, they need carbon, every living organism needs carbon. Okay? But we can organize them based on where are they getting their carbon and where are they getting their energy source. And if we do that, then we can have a good idea of where we're going to find them. So yeah, carbon makes up 50% of the composition of the cell. That's for most living organisms. And so we have bacteria that are called chemoautotrophs, and I'll show you what that means in a minute. But we could know because of what they metabolize that the only place we're going to find them is where you have a significant amount of carbon dioxide and a significant amount of inorganic material for energy. And the only place you're going to find like that are hydrothermal vents at the bottom of the ocean. Okay? And so we can know where do we go looking for these bacteria. Or let's say our bacteria are photoautotrophic. What does photo mean? Light. And so you're like, now they want light. Where do you find light? Well, where there's sunlight. And so we know where are we going to find these type of bacteria, where there's ample sunlight. Yeah. And then this is a byproduct of this, but because prokaryotes are basically found everywhere, which means that they're ubiquitous, and because they have a number of different metabolic strategies, you just find bacteria literally everywhere playing a huge role in cycling these nutrients around. They need these nutrients just like we do. You find them basically everywhere, in soil, in water, the bottom of the ocean, 50 feet underground, in these oil deposits that we're sucking oil out of for fossil fuels, right? You just find bacteria everywhere and playing a huge role in moving these nutrients around, right? Because I know what you're wondering. You're sitting there and you're wondering, okay, I need these nutrients just like bacteria do, but I'm huge, right? Compared to a bacterium. I mean, I'm huge, period. But we're compared to a bacterium, like we're huge, right? You're like, wow, that's a lot of material. And then, I don't know if you knew this or not, but there's a pretty good probability we're all gonna die, right? Let's get really depressing here, right? And you're like, wow, we've got all these nutrients just stuck in a body that's not doing anything, right? And it's like, how do we free those nutrients up? I know you're sitting there, you're wondering it. There's a pretty good probability that there are bacteria that are already living all inside you, all over you, that they'll just eat you when you're gone, right? And they'll recycle those nutrients and make them available for other organisms. Just because you die doesn't mean the bacteria in your gut stop eating. They just keep going. They just keep eating. All right, so this is a table from the textbook, and it's horrible. This is a horrible table. And so I put it up here just so I could show you it's a really bad table, because it's like tables should be very easy and intuitive to read. But it's like we've got two different rows of headings, and it's like, which heading do I read? It's just horrible. It's a horrible table. So here's the same information, but in a much better table. This is a much better table. This is not from your textbook, but you have access to these slides, so now this is resources that are part of who you are. Meaning, I expect that that you've seen this, right? And I just showed it to you. Although Karsten left us and some other people aren't here, so they didn't see it. But anyways, all right. So over here on the left, it shows us where are we getting the carbon. And this, I say we, I'm meaning the bacteria. We're not bacteria. But where are the bacteria getting their carbon? Although you could put any organism in here. And then where are they getting their energy? Carbon, energy. And if their carbon comes from organic molecules, then they are a heterotroph. Okay? If their carbon comes from carbon dioxide, they are an autotroph. Okay? That's cool, right? A very easy table to read, right? Where does the carbon come from? It comes from organic molecules, heterotroph. If it comes from carbon dioxide, autotroph. And it's like, where do they get their energy? Do they get it from oxidizing molecules, or do they get it from light? If they get it from light, they are a photoautotroph or photoheterotroph. If they get it from oxidizing molecules, they are either a chemoautotrophs or they are a chemoheterotroph. Okay? Where do you think we fit on this table? We are chemoheterotrophs. We get our carbon from organic molecules in the food that we eat, and we get our energy from oxidizing those molecules from the food that we eat. We are chemoheterotrophs. Okay? What about a photosynthetic bacteria? Okay, so it's photosynthetic, so it's getting its energy from sunlight, and then it's going to be in one of these two categories. And the answer is, you have bacteria in both. You have some bacteria that are heterotrophs. They get most of their carbon from eating other things, but then they get their energy from sunlight. You're like, wow, it seems selfish that they're using sunlight for energy and then still eating other things. It is a little selfish, but it's okay. I mean, for them. So here's just a diagram from the text. This one's not an awful diagram. Just showing you that this diagram and the next one, they're just helpful reminders that bacteria, bless you, play integral roles in recycling and moving nutrients around ecosystems. Okay? All right, let's do something less depressing. Let's do a big, giant redwood dies. You're like, that's pretty depressing. But it's not you dying, right? And so you've got like a 350-foot tall tree that's, 6 feet, 8 feet in diameter at the base. That's a massive organism with a lot of carbon, a lot of oxygen, a lot of hydrogen, a lot of nitrogen, a lot of phosphorus all inside of it, right? How does that get unlocked for other organisms to use? It's the bacteria. They decompose that tree, bacteria and fungi, and they unlock those nutrients for the plants in the soil around where that tree fell. Yeah.
I've also heard that like sometimes the fire as it's burning dead trees or dirt voids can also host?
Yes, absolutely. Fires do a wonderful job of unlocking nutrients. Because fires, you get basically the carbon molecule reacting with the oxygen in the air to produce carbon dioxide and water. Okay, but everything else just kind of falls out. And so the nitrogen and the phosphorus and some of those other minerals, they just fall out. So that is true. Fires do a wonderful job of unlocking nutrients. And they do it quickly, right? Anyways, sorry. All right. Man, this has been the best Friday ever. I don't know about you, but it's like, man, I woke up this morning, I knew it was going to be a good day, and it's meeting every expectation. That's what I want to say. It's meeting every expectation. Okay? Gosh. So our next question, now we're going to start to deal with more specific questions. How do antibiotics contribute to the problem of bacterial infections? Because I know this, when we started talking about bacteria, your first thought is you're like, how do we explain antibiotic resistant bacteria and what can I do to help the global problem, right? Anyways, so thousands of human diseases are caused by bacteria. Basically, as old of human writings as you can find descriptions of pathology that we know are caused by bacteria. And then you've got groups of organisms that were really good at actually preserving their humans that died. The Egyptians were really good at that, and even the Greeks. They wouldn't embalm like the Egyptians would, but sometimes they would do a really good job of burying them, so they'd be pretty well preserved. So on top of reading the history, and you're like, I'm pretty sure I know what that pathogen is, you can find bodies and pull DNA out of them of bacteria. And so we know that the plague of Athens that happened in about 430 BC that killed a quarter of the Athenian army was caused by typhoid fever, but that bacterium is Salmonella enterica. Yeah.
This is like a weird question and probably just sounds dumb, but like when we die, if we died of a disease,
Yes.
just like plant diseases can affect us, can human diseases affect like the ground or
Some of them, yeah. No, that's not a dumb question. Some of them. I mean, if they are a human-specific disease, no. If they are a mammal-specific disease, they might impact only mammals. But there are diseases that are more ubiquitous that impact everything for sure. Yeah, that's not a dumb question. Bubonic Plague killed nearly half of Europe over two different periods, 541 to 750. Again, 1350-ish to 1360. Yersinia pestis, and this is like a gift that keeps on giving. Every year there are about 5 to 20 cases of bubonic plague in just the United States. Worldwide, I don't know what the numbers are, but this is a bacterium that just keeps coming back, keeps cropping back up. On top of this, we have new diseases that we discover every year that come from animals. Diseases that come from animals are known as zoonoses. And so the animals that we love to keep around us, they love to share their bacteria with us. Either when we eat them, or when we pet them, or when we drink water that they've been drinking in or defecating in, right? They just love to share their bacteria with us. And so here's now the answer to this question. So we tend to really like to use antibiotics in agricultural applications, right? Like, we don't want our cattle dying because then that raises the cost of beef. So we'll just treat them all with antibiotics, right? Even if they're not sick, we'll just treat them with antibiotics regularly to keep so that they don't get sick. Okay? And then, if you've got an ear infection, who knows if it's caused by a bacteria or not? We'll just give you antibiotics, right? Right? We'll just treat it with antibiotics just in case it might be a bacterial infection. The problem with this is the more you use antibiotics, the more likely it is that you're going to develop antibiotic-resistant bacteria. And so your target bacteria may not be the one that develops the resistance. It may be some other bacteria that just happens to be in the same place. And in the presence of this heavy selective pressure for antibiotic resistance, you get antibiotic-resistant forms. So some examples of this: methicillin-resistant Staph aureus, Staphylococcus aureus. This is MRSA. Okay, it's probably the most well known and well studied of all the antibiotic resistant bacteria. Vancomycin resistant Staphylococcus aureus. Versa, sometimes this is vancomycin immune, so that it's Visa, right? Because Visa sounds better than Versa. Right, Versa, that's a a cheap car that Nissan makes, but Visa, that's a credit card, right? I like that. And so because of this, and because of our inability to fight these antibiotic resistant forms, a number of microbiologists actually suggest that we are poised and ready for a global pandemic, for the spread of just a massive antibiotic resistant strain of something that will just travel the globe. Now, luckily, these antibiotic resistant strains tend to be less virulent, means they grow less quickly. They're not as infective as the non-antibiotic resistant forms, but we can't treat them. And so your MRSA infection may not spread as quickly as a staph infection that is not methicillin resistant. But still, I mean, it's like, how do you treat it if you don't treat it with antibiotics? I mean, you treat it with other antibiotics, right? You make new antibiotics, or you just cut it out, right? Just scoop it out with like a melon baller. Yeah. Man, I'm building some wonderfully vivid pictures for myself. These are just coming to me, so I'm sharing them. Yeah, Naomi.
I had MRSA for like 4 years.
Yeah, and it's just, I mean, it's,
Interesting.
so I mean, for 4 years, you're trying to eliminate something that if it wasn't antibiotic resistant should be gone in 3 or 4 days.
So I literally took like, I took meds like every 4 hours, so we just like.
And probably 15 different antibiotics over those 4 years.
That's very interesting.
Yeah.
Would that make you more likely to get MRSA again?
Maybe, but probably not. Yeah. But I mean, using other antibiotics makes it more likely we'll develop Staph aureus strains that are resistant to those new bacteria, these new antibiotics. Sure. And so here's just some pictures of some of these. Here's Salmonella enterica, the causative agent of typhoid fever, killed a quarter of the Athenian army. Here's Yersinia pestis and somebody with Black Plague. And then, just to kind of show you what's going on, here is a map of all of the emergent diseases. So these are antibiotics that we've been able to treat since like the 1950s, when antibiotic use really became popular and easy. And then they're recropping up, even though they're bacteria that for 50 to 60 years we had no problem fighting, but now they're antibiotic resistant. And so they're coming up and then they're spreading. So any of these could be the source of the next global pandemic, right? So this is a map where you just look at it, and so that way if 5 years from now we're in the midst of a global pandemic, you can remember this map and say, hey, I remember where that came from. Remember where it came from. Oh, and then just some more fun pictures. So this is Lyme disease. This is another bacterium. This is a spirillus bacterium. You see that spiral shaped? This is Borrelia burgdorferi, the causative agent of Lyme disease. Here's the bullseye lesion that's characteristic of Lyme disease. And here's the life cycle of Lyme disease. And then let's just keep going. Yeah, did you have a question, Allison?
I was just wondering, why is Lyme disease so hard to detect? Like, I've known friends who had it and it took them years to figure it out.
Yeah, it's because the bacteria hide out, and so it's really difficult to get a positive result of the bacteria. They're really, really small. I mean, all bacteria are small, but these are really, really small. These are like somewhere between a typical bacterium and a virus in size, and they usually hide out. So, I mean, it can take years or decades to get a positive result. So false negatives are really, really common with this. And they usually don't like to treat it unless you get a positive result, because the most successful form of treatment is to put somebody in a hyperbaric chamber and just load them up with enormous amounts of antibiotics. It's very expensive. You know, it's like you either have to send them home with a hyperbaric chamber or keep them in the hospital. So, yeah, if you can catch it early, it tends to be easy. Like if you've got that bullseye lesion, you took a picture of it, then they'll just treat you with antibiotics then. They'll usually take care of it. But if you don't catch it then and you don't always get the bullseye lesion and you actually have a significant number of bacteria, the treatment is just really, really complicated. Yeah.
I was gonna say it's also kind of hard to catch because I know a few people who had Lyme disease and like the symptoms in the beginning are like just like a cold or
Sure. Yeah. Yeah, it's often misdiagnosed. Yeah, Mika.
Is there any particular reason why it's a bullseye?
Yeah, that's it. I mean, it's just the pattern of inflammation. That's what it ends up doing. Yeah, that's a good question. So, all right, one more image, and then we'll be done for the day. This is Staphylococcus aureus, and this is MRSA. Although it looks just like non-methicillin resistant Staphylococcus aureus, unless you look at it genetically, but morphologically they look the same. All right. Well, happy Friday. Enjoy the rest of your Friday. Hold on to those vivid images in your minds, and we'll see you on Monday.